Hello students, so today we'll be discussing about the topic plant growth regulators, which is a chapter of class 11th biology. Okay, so what are plant growth regulators? As the name itself is suggesting, the regulators, the chemicals, the compounds or the substances which regulate, which control what? Which control the growth of a plant. Okay, so basically apart from your food, your oxygen, your temperature, there are certain chemicals or you can say hormones that are produced inside the plant only, and those hormones regulate or monitor the growth of a plant. So these are known as your plant growth regulators. Now regulators can be of two types. Regulators can be growth promoting and they can be growth inhibiting. Growth promoting and growth inhibiting, both the categories or both the types are necessary for plant. Why? Because not only the growth is important, but after a certain time, after a certain period, the end is also necessary. Things which takes birth, things which take birth or things which come into existence need to be finished also, so that it can be replaced by something new and better. This is the law of nature. So when something is growing in plant, that needs to come to an end also. So for that, inhibiting factors are also there, or the process that has been started that needs to be slowed down at time at certain phases of plant's life. So the inhibiting hormones are also there. Okay, now in category of growth promoting hormones, we have auxins, we have cytokinin, and we have gibberellins. Okay, and in category of growth inhibiting, we have ABA, that is abscisic acid. Abscisic acid. And the fifth one is ethylene. Ethylene acts as both. It can be a growth promoting hormone also, and it can be a growth inhibiting hormone also. So ethylene comes in the midway. It has dual functions. Okay, now we'll be starting with the general characteristics of plant growth regulators. Okay, characteristics. Plant growth regulators are also known as PGRs in short, that is plant growth regulators, or these are also known as plant growth hormones. Plant growth regulators or plant growth hormones, which are synthesized by plants themselves. Okay, so talking about the characteristics, the very first thing, obviously when we are talking about growth, the growth directly signifies what? Cell division. So the basic characteristic is going to be cell division. Then second, it or they promote new organ formation. Organ means new leaves, new fruits, branches, etc. Okay, so first they'll be promoting cell division, then they'll be promoting formation of new branches, new leaves, new fruits, etc. Then third, they are responsible for aging of plants. Aging means when plants enter into its senescent stage, slow aging. Then they control many physiological processes like respiration in plants, then transpiration, etc. Okay, then they can be responsible for inducing flowering, can be responsible for dormancy. What is dormancy? Dormancy is the stage where your seed is not germinating, where your seed is in resting phase. Dormancy is not the dead phase. Dormancy is a resting phase when the seed is performing its basal metabolic activity but is not showing any signs of growth. It is not dead, it is living, but it is not growing. It is carrying only respiration and basic metabolic activities to maintain its state of living. That's it. And once the favorable conditions of temperature, pressure, atmospheric air, or nutrients in the soil becomes favorable, then this dormancy breaks and seed germinates. Alright, so dormancy is also controlled by these factors. Dormancy can be induced or broken down by these factors. So we'll be talking about one by one about all these factors. First PGR is your auxin. Okay, so auxin was the first PGR that was discovered. After the discovery of auxin, then later on other PGRs got discovered. So auxin holds the first position. Who discovered auxin? It was discovered by Charles Darwin, and his son Francis. Charles Darwin and Francis Darwin. Okay, what they were doing, they were studying an experiment of theirs where they saw the bending of coleoptile of canary grass. What they observed? Bending of coleoptile of canary grass towards light. So what they noticed, what is coleoptile? Coleoptile is the protective covering of plumule in a seed. Which seed? Monocot seed. Coleoptile is what? This is important. Coleoptile is covering of plumule in monocots. Protective covering in monocot sheath. Plumule is what? The structure that gives rise to future shoot. Okay, so they observe this bending of coleoptile towards the source of light. So they concluded that there is something what they concluded, that there is something. They didn't name that chemical, they just said that there's something which was responsible for the bending of coleoptile towards light, which they termed as phototropism for the phenomenon. Later on, that something was isolated and named as auxin. Named as auxin by F.W. Wendt. Okay, so discovery was done by Darwin and his son, and later that something which they observed was isolated and identified by F.W. Wendt. Okay, now coming to the main functions which auxins carry. Functions of auxin. The very first thing, auxins show apical dominance. Now, what is apical dominance? Apical dominance is that they are present in the apex, at the apex of plant. And what is the meaning of apex? That they are present at the tip of root and shoot. Okay, so they are present at the tips, that means at the topmost part of the shoot and at the lowermost part of root. So what they do, they are responsible for increasing in the overall length or height of your plant. Show apical dominance, that means they are present at root and shoot tip. Okay, they are present at the root and shoot tip of plants and responsible for increasing overall length of a plant. Second, they induce cell division. They induce cell division with the help of, in combination with your this cytokinin. Okay, cytokinin is your other PGR, which we'll study later. Then they induce flowering. They induce flowering in, obviously, flowering plants. Then they induce root initiation. Root initiation where? Root initiation in stem cuttings. In stem cuttings means the stem cuttings in your plant tissue culture laboratories, where we try to grow plants artificially in a laboratory conditions. Okay, so there in a stem cutting, if we have cut a stem and provided a particular nutrient medium with auxin in it, auxin will induce the rooting in that stem cutting. Okay, then it also acts as herbicide. It acts as herbicide. What it does, it kills dicot weeds. Acts as herbicide and kills your dicot weeds. Then one more important point, it induces parthenocarpy. Now what is parthenocarpy? Parthenocarpy is formation of fruit. Parthenocarpy is formation of fruit without what? Without fertilization. Okay, So these were the major functions of auxins. Parthenocarpy is also important, where fruit is formed without your fertilization. Okay, then next important was your apical dominance, that it shows it is present at the tips and thus is responsible for lengthening of roots and your shoot tip. Then next hormone, next PGR is your gibberellins. It is also denoted as GAs, gibberellic acid. The first gibberellin was extracted as gibberellic acid. By whom? By Kurosawa. Okay, it was discovered by Kurosawa, Kurosawa, where in false Bacon disease. Wait. So the gibberellins were first discovered, not isolated, discovered by Kurosawa, and then isolation was done by Kurosawa, just discovered them, and then came your isolation and extraction. Extraction and isolation was done by Yabuta and Sumiki. There were two other scientists, Yabuta and Sumiki, which were responsible for the isolation and extraction of this gibberellin. From where did they extract this? From rice of false Bacon disease. Now, what is false Bacon disease? False Bacon disease was a disease in Japan where rice seedlings, rice pods were suffering from the infection of a fungus. That fungus was known as Gibberella fusicori, and that fungus contains these chemicals. So from the DNA of that fungus, it was extracted by Yabuta and Sumiki. False Bacon disease basically was the rice pods which were hollow from the inside. From outside they looked all perfect in shape and size, but from inside they were all hollow. Okay, so coming to the functions of gibberellin. What does gibberellin do? First, the very first thing, gibberellin induce flowering. These are the positive promoters or the promoters for flowering. Second thing, they increase length of internodes. Increase length of internodes, that means they overall increases the length of plants. So these are basically used by brewing industries like sugarcane, where this gibberellin is externally sprayed on the sugarcane, and thus the sugarcane stem elongates more, so that brewing industries get more product. Third is your bolting. Now, what is bolting? Bolting is again the internode elongation in rosette plants like cabbage, and internode elongation plus flowering. Bolting, if suppose this is your cabbage, leafy structure, compact, and if we'll spray gibberellin, the leaves will open up like this. It will elongate, and at the tip it will induce flowering. So this particular thing is known as bolting. Why this bolting is necessary? This bolting is necessary to induce your flowering stage in plants, and thus you can get seeds for the next generation. Alright, then third is it breaks seed dormancy. The very important factor is that it breaks seed dormancy. It causes the germination of seed once the favorable conditions have been attained. Okay, then next will be your, after gibberellin, cytokinin. Cytokinins, these are the third PGRs. Cytokinins were discovered by Skoog, Miller and co-workers. Okay, cytokinins were discovered by these three scientists and their co-workers. Cytokinins were previously known as kinetin. Okay, what does cytokinin do? Cytokinin is responsible for, if we talk about its function, cytokinin is responsible for breaking, again, seed dormancy. Since it is also a plant growth regulator, promoter, so it will be responsible for breaking of dormancy, continuation of flowering, delaying of senescence, plus in combination it promotes like apical dominance was seen in the case of auxins. Cytokinins are responsible for lateral growth, promotes lateral growth in plants, or it can be formation of lateral buds or branches, etc. Okay, then it also induces root and shoot formation. Induce rooting and shooting in plant tissue culture. In plant tissue culture, depending upon the ratio in combination with auxin. Okay, cytokinins plus auxins ratio vary, and these varied ratios are responsible for formation of root and shoot. Okay, then they delay senescence or they delay aging in plants. Fine. Then your fourth hormone or fourth PGR. Fourth PGR is your growth inhibitor, your abscisic acid. Abscisic acid. Now we know the name itself is telling its function. Abscisic acid means it is responsible for abscission. What is abscission? Abscission is to hold, to restrict something, or to slow down certain processes. Which processes? The normal physiological activities whatsoever are being carried in your plants. These all are restricted by this abscisic acid. Okay, it was first isolated by F. Edicott. So F. Edicott was a scientist that first isolated your abscisic acid. Now what he did, what he noticed was abscisic acid was responsible for majorly the negative or the growth retarding factors. Like it was, no, if you suppose, if you are confused what to write in the functions of abscisic acid, and if you are well aware of the functions of your cytokinins, your gibberellins and your auxins, just write the anti of all of them in this section. Abscisic is, you can also write it as, abscisic is the anti of all PGRs. Okay, which PGRs? Growth promoting PGRs. So what this abscisic acid is doing, first thing, it promotes senescence, okay, rapid aging, then it promotes dormancy, it will create, then early fall of fruits and leaves, then closing of stomata, if stomatas will be closed, then obviously your transpiration and photosynthesis will drop down. Then fifth is your ethylene. Ethylene was discovered by scientists called H.H. Cousins. Okay, what they noticed was that there is some volatile substance. Volatile substance which is coming out of ripened fruits. And is responsible for the ripening of unripened one. Okay, so what he concluded was that this ethylene, it was responsible for ripening of fruit. The major function is fruit ripening. All right, but before that, one more thing is there. It is the only PGR that is present in gaseous state. All other are crystallized compounds. This is the only PGR that is present in your gaseous state. Okay, now first thing, the function. So its first function is it promotes flowering in mango. Then respiration in plants. Okay, then fruit ripening and then dormancy, responsible for breaking down of dormancy. Respiration, it slows down the process of respiration. Here it is showing negative function, here it is showing positive, here also it's positive. Dormancy, it helps in breaking down of dormancy. Okay, then sometimes with the help of or in combination with AB, it is also responsible for closing of stomata closure. So this is also going to be your negative function. That's why we were considering this ethylene as a combination of both growth promoting and growth inhibiting hormone. Okay, so this was the general introduction of PGR. Of course, there is a lot more detail about each of them, how the discovery happened. I have just mentioned you in short that who discovered what, who isolated these things, what are the major functions, but there is a lot more to study in this section. However, for now, this is more than enough, and the questions from this section could be asked as in the name of scientists, or the discovery, or apical dominance, lateral dominance, function of cytokinin, ethylene, your abscisic acid, which are PG promoters, which are your inhibitors, what is bolting. Okay, so yeah, this all. And one more thing I was recalling in case of cytokinin, yes, the functions, one more function you can add, that it is responsible for cell division. Auxin was also there, but main is this cytokinin. This induces cell division, where in normal plants also, and in case of your plant tissue culture also. Okay, so this is all about your PGRs, the general introduction. Hope you have understood a bit from this section and have benefited. All right, thank you.